Hello, everyone, and welcome to a Quality Perspective Roundtable discussion on the successes and challenges of making changes to the pharma workflow. My name is Mike Auerbach. I'm the editor-in-chief of American Pharmaceutical Review magazine. This roundtable discussion will focus on challenges faced by pharmaceutical quality teams in their material analysis workflows and real-world solutions. How leading pharmaceutical manufacturers have adapted their quality processes to include handheld Raman, and what areas within their workflow leading manufacturers use handheld Raman. With us today are Jill Carrero. She'll be our moderator. She's the Global Business Development Manager for Rigaku Analytical Devices. Dr. Michelle O'Connor, Senior Scientist at Lindra Therapeutics. Dr. Joseph Stoltz, Senior Principal Scientist at Pfizer, Dr. Mark Levine, Senior Staff Scientist at Genzyme, Dr. Suzanne Schreier, a Senior Application Scientist at Rigaku Analytical Devices, and Dr. Valerian Shibata, Application Scientist also at Rigaku Analytical Devices. In addition, this event will hold a live question and answer session at the end of the presentation. To ask a question, click on the Ask a Question tab on the right side of your screen. Please take note the right side of your screen also features an overview of this webinar and more information regarding our speakers. We'll also be holding polling questions during the presentation. When a polling question appears on your screen, please answer the question, and our panelists will discuss the results. If you have a technical question during the event, click the Test Your Connection button at the bottom of your screen, and that will give you more access to webinar help. Finally, we encourage you to use the social media widgets beneath the webinar to share with your friends and colleagues. Now, I'd like to hand the discussion over to Jill Carrero, who will be the moderator, as I mentioned, for today's discussion. Jill?
Thanks so much, Mike. We really appreciate American Pharmaceutical Review for hosting us today. Thanks to all of the attendees for joining this Rigaku Roundtable, and certainly huge thanks to our panel. We hope this will be a really awesome conversation between some of my very favorite quality-minded pharmaceutical professionals. I'd like to take a minute to introduce you to our company, get you acquainted with Rigaku. We were founded nearly 70 years ago in Japan, and we've been making analytical tools ever since. We formed around the first commercially available rotating anode X-ray generator, and you very likely know Rigaku from maybe our XRF or XRD products in your laboratory today. That is Yoshihiro Shimura, forgive me, who founded our company in 1951. And his son still leads us today as our CEO. His name is Hikaru. We have locations all over the world, laboratories and offices both, headquartered in Tokyo, Japan, but we also have 3 major facilities here in the United States, including one in Massachusetts, which is also where I'm from. So Rigaku Analytical Devices, there in Wilmington, Massachusetts, is where we make both of our handheld Raman devices and our handheld LIBS devices. We have a CE or a ISO 9001 certified facility there, which is where we grow our clean room grown lasers. And of course, our instruments are built to be used. They're CE compliant and MIL standard rated. So that essentially means a lot to our military customers, but for you and I nowadays, it means that you could give your progeny a bleach bath if you so chose to do so. Most importantly, we're a pretty unique team in that there are about 50 of us located in that facility, all focused on handheld spectroscopy. And I have the pleasure of having a couple of my colleagues here with me today to answer your questions. We started about 10 years ago with the introduction of Progeny in 2014. That's our 1064 nanometer based handheld Raman for pharmaceutical business. And then not too long thereafter, its brother for the military and police applications, the Progeny Rescue, was introduced, along with Katana, our handheld LIBS device. And over the years, we've been proud to be awarded a number of great R&D innovation awards, including one just recently in 2020 from Security Today for our Sequel line of products, which is really cool combination between colorimetrics and Raman, which allows us to do some trace level detection that usually requires SIRS and things like that. But today we're here to talk about Progeny, which is our handheld Raman tool for the pharmaceutical environment. So I'd like to delve into that just a little bit. Starting with, what does Progeny tell us, right? Simply put, it's a verification tool for raw material identification. Progeny will compare a reference spectrum and a sample spectrum and provide pass or fail results. And the screens on a Progeny look like that, pass or fail. That difference is the result of an algorithm threshold that is set by your team during application development. And if Progeny doesn't positively identify your material, well, then it'll search the onboard library of 13,000 items to figure out exactly what you've got at hand. The goal of implementing a tool like Progeny, well, it's pretty simple. It's to perform the required ID testing that the FDA or other regulatory bodies say we have to do, and to do it as effectively, efficiently, and safely as possible. That's the goal. So what's our goal for this time with you? It's a little bit different in that, we know how many folks have been spending time on Zoom and webinars. So we wanted to do a little bit something different. We wanted to learn together with you. So let me introduce you to our panel, because I trust them and I'm sure that you'll learn a lot today from them. They're from a few different companies here in the United States. Then Lindra Therapeutics, which is based in Massachusetts. Sanofi Genzyme, also here in Massachusetts, a site, and then Pfizer, which is in Groton, Connecticut. I'm going to introduce to you first Michelle O'Connor, and you see Michelle here with me. Hi, Michelle, hiding behind your mask. Yep. I do want to say we have some technical issues in having one of our attendees on the line, but Michelle, if you wouldn't mind telling us a little bit about yourself at Lindra and the work that you do.
Sure. Thanks, Jill. And thank you so much for having me. So I'm Michelle O'Connor. I am a senior scientist in the Analytical Research and Development Group at Lindra Therapeutics. I've been in the biotech pharma industry for over 10 years, working in analytical development. I have experience with characterizing small molecules, polymers, mRNA, DNA, viral particles, a wide variety of APIs and excipients. And right now I am working on building the quality control group at Lindra to support efforts to commercialize our products. So I'm also involved in some of our building capacities for real-time release, in-process control, and QBD. The work that we do at Lindra with Progeny is mostly releasing raw materials. So we have many different types of polymers and excipients that we use in our dosage form, which you can see on the right-hand side of the screen. And we also have release testing of different APIs. We also use the Progeny for capsules, capsule coating, and polymorphic form assessment, both at release and in stability. We use the Progeny for ID testing, for in-process intermediates, and for our finished products. So we actually use the Progeny kind of at every point in our manufacturing.
It's so cool too. And then the product that you make is really awesome. I'm excited for you to share a little bit more about that in just a minute. Thanks, Michelle.
Yep. Mm-hmm.
Joe, I see you there. Thanks for joining us. This is Joe Stoltz from Pfizer in Groton, Connecticut. And Joe and I have had the pleasure through his work as a principal scientist at Pfizer for 20-something years. We've worked together for over a decade. And so, Joe, if you wouldn't mind sharing with us a little bit about who you are, but mostly what you do with Progeny very generally.
Yeah, I've been at Pfizer in Groton. I've worked in commercial manufacturing, and I work in analytical research and development. So we use a whole bunch of different Ramans. The Progeny in particular we use for API raw material identification, excipient identification, post-packaged identification of packaged clinical drug products, And we also use it for the occasional in-process control testing applications during API synthesis if we're looking for some particular polymorphs. And most recently, we've applied it to functional excipient testing.
Super cool. And you're going to share with us more about that in a little bit too. So thank you, Joe.
Yeah.
I'm very grateful for you being here. So then I turn it over to Mark, who we don't have, unfortunately, on camera today, between all of the firewalls and VPNs that are always changing around in the pharmaceutical world, unfortunately. But I do believe, Mark, we have you on the line. This is Mark Levine from Sanofi in Framingham, Massachusetts. And Mark's been a principal scientist at Sanofi Genzyme for over 28 years. And you bring a unique perspective to our panel today, Mark, in that you come from the process development side of the world.
Yeah, that's right. I've mainly worked in process development for most of my career. More recently, I've been in clinical trial manufacturing at Sanofi. And I was asked about maybe 4 years ago to start a raw material identification program for the materials that are coming in for our clinical trial manufacturing group.
And then so they just kind of handed you a progeny, right, and said, go for it. But can you tell us a little bit about, certainly you do some polysorbate work with it as well, right?
Yeah, that's right. So we're using it for about, I think maybe about 80, it identifies about 85% of the raw materials that we use for the various programs in the clinical trial manufacturing group. But one of them was polysorbate 80.
Okay.
We were able to do a test just using a Raman, which could distinguish between polysorbate 20 and polysorbate 80. And we were able to do that test through the shipping container that the polysorbate 80 came to us in. And that was important for us because it shipped to us under an inert atmosphere. So we had to open the bottle to sample it. We'd be faced with using degraded material in the manufacturing.
Yeah, you start that timeline, right? That's a pretty important application for a lot of people. I'm glad that you guys had success with it, Mark.
Yeah.
And this is exactly why, right, guys? This is why thank you, Mark. We've asked these folks to join us so that you could hear it directly from the horse's mouth, so to speak. They're all definitely cool folks, but they're quality-minded, but most importantly, they've used a bunch of different tools like the one that we make for raw material identification. So I want to ask then a question of our attendees, if you don't mind, Mike. Can we ask our attendees a question now, a little bit of a poll question?
Our first poll: what technology does your site use for its bulk raw material identity testing? So your choices are portable Raman, benchtop NIR, FTIR, an outside lab does it, HPL, GC, wet chemistry, etc. I'm not sure, and this doesn't apply to me. So please, everyone, make your selections. We'll give everyone a few moments. And what we'll do right now is we'll gather all of our results, and hopefully everyone can see the results now. A little nice little pie chart there. So we've got about 45% with portable Raman,
Okay.
about 9% with benchtop, HPLC, GC wet chemistry, 9%, and not sure, about 36%. So if Jill, I'll hand it back to you in case you want to make any comments regarding this.
It doesn't surprise me. The world has joined forces with handheld Raman because it's the most efficient way to do a job that we really have to get done. And so I do want to talk just a moment about how handheld Raman changes the workflow, and I'd like to take a look at this figure because it captures it pretty well. Specifically on the left, we have how kind of the raw material identification testing flow works in a traditional way. And then on the right, we have how it's streamlined using a handheld Raman. when we take a closer look at the traditional workflow, we see a couple of really time-intensive, personnel-intensive areas. Most specifically, when we focus in on these offline lab processes in red, we really see where the bottleneck happens. That's where the traditional flow is really focused around kind of dependency on the quality lab and the turnaround time. And so when we take a look at how the traditional flow works, we're really implying a lot of opening of containers, moving product around, using that sampling suite. But more than anything, it's about time. It's about waiting hours, days, sometimes even weeks to be able to release product. And when we move to this flow using a handheld Raman, we see essentially all those red steps are eliminated because we minimize the amount of sampling that we have to do. Because using a handheld Raman, we test the material in the container of receipt. And so what that allows us to do is really maximize the throughput of our systems because we're doing everything in the warehouse. And what that also allows, of course, is minimized risk of contamination and personnel contact. And we just increase productivity exponentially when we put in a handheld Raman into work. But I really did want to know, how true was this, right? How valid was this? So I began by asking our panel, where was this accurate? And they gave me some feedback. There is no doubt about it. When first and foremost, where we were accurate is that, the priority definitely continues to be minimizing sampling, right? We want to certainly make sure that we're reducing exposure and sampling as much as we can. And that, electronic data management is really a key goal of everybody. Unfortunately, in the complex and really slow environment of the pharmaceutical world, what we still continue to see is some sampling, no matter what. There is always some sampling that's required of the system. And we also see kind of a carryover of paper processes continue on. And even 10 years of doing this, I still see a lot of paper processes that sometimes surprise me in this electronic world. But when we talked about this, Joe, you gave some really, really interesting feedback to me that I'd love for you to share with our audience about kind of how the nature of pharma has shifted from being much more siloed. It used to be much more siloed into what it is now and how this kind of affects your team and how it affects the jobs that you do around this. Would you mind sharing that with the audience?
Yeah, sure. So what I meant by siloed was we used to have an API raw materials release group. We used to have a drug product stability group, API release group, drug product release group. And now we operate under the model where all of those functions are combined into one group. So analysts do stability testing, they do clinical release testing, they do excipient testing, they do raw material testing, and they even do in-process control testing. So that's what I mean by not as siloed anymore as we once were.
You're all jacks of all trades now.
Right. You have to be very flexible.
Yeah. And I'm sure that even, as the pandemic situation has arisen, that's driven that even further, I would guess.
Yeah, for sure. Yeah.
And Michelle, when we were talking about this, you brought up something that we see all the time too, is that you're getting there, right? You're not to FDA, full approvals yet for human release. So you're kind of moving in this direction, right?
Yeah, absolutely. I think this is a good goal for us, but we're a young company and we don't necessarily have, a doc with a dedicated person there to kind of do material ID and receipt. So, it's not quite as streamlined as this flow chart would. And that, like I said, that is our goal. And I think the Progeny makes that much more possible.
I hope so. I hope it's a big part of your success. That's for sure. So that said, we know companies like yours, lots of companies see real gains from using Progeny. Mainly cycle times improve massively. When it comes to personnel, we see within organizations the ability of personnel to shift to more priority tasks because they're not doing, run-of-the-mill RMID kind of analysis work. From my perspective, the biggest gains that aren't measurable come in the forms of kind of analytical support of other areas. That happens all the time when using a tool like Progeny, specifically because it is such a versatile, handheld Raman. But more than anything, and I see this a lot, especially with the customers and their teams that are on the line today, is that there is an important connection that can be formed between the engineering group and R&D. As formulations are developed and we go through the process of kind of figuring out what works and what doesn't work, Progeny can become a communication tool in that aspect. So those are some of the real gains that we see organizations kind of find. But I wanted to, again, turn it back to our panel. You've used different handhelds. Joe, Michelle, I know you've used a bunch of different handhelds. What about Progeny, or Progeny as you say, is best for the work that you do?
I can go first if you want, or
Go for it.
okay. First of all, so we've used 785 nanometer instruments, and we have the 1064 instrument. As far as opening up the field to more compounds that you can analyze, I do like the 1064 a lot better because of the less fluorescence that you get with the 1064. But the other thing I like about the Progeny is the software as it relates to data integrity, Alcoa, and those type of principles that you're operating under in a GMP environment.
Data is God, right, Joe?
Absolutely.
What about the spectra? Forget the spectra. Who wrote the spectra? Who took the spectra?
Right. Who logged on to the instrument? Is it clearly identifiable who they were, what they did when they were there? Is it all captured in an audit trail that's easy to read and sort through? But the other cool thing is, I can remote into the Progeny that's sitting at work, or anybody else can, to review the data. So that's pretty cool.
Cool. I'm glad you can, again, make use of that nowadays, right? Michelle, you have some things I think that you kind of like about Progeny as well, right?
Yeah, I think just to echo what Joe said, the 1064 laser is just it's a vast improvement for me in terms of the level of fluorescence, like you said. I think it's just unique in that way. And then the other thing I really like about the Progeny is the ease of use and how quickly I can train people to use it themselves. So, it's quickly become a tool that many people at our company use. And like Joe said, it's very easy to see who was using it, so you can really keep the data integrity part as well.
Cool. And then Mark, I know you're on the line. Thank you, Michelle and Joe. Mark, I can't see you, but you in your role in process development at Pfizer, you weren't kind of on the inventory or QA side of things, but you had a unique experience in that they came to you and said, here, set up a raw material identification program, right? Can you tell us a little bit more about that?
Well, I didn't actually have, very much experience with analytical methods, except, doing them on the level where you'd be involved in, like, release raw materials or products. But I guess there was no one else available to do this, so that's how the project landed in my lap.
Happens more than you'd think, I think.
Yeah, and I had I had been involved in a project to evaluate a 785 nanometer instrument, and the quality of the data was like so poor, and the amount of fluorescence with the raw materials we were looking at it made the instrument unusable. I mean, we never advanced it to the point where we would actually use it for release of raw materials. But when we got your instrument, it worked immediately from the start. We were very quickly able to be confident that it was capable of doing identity testing on like a lot of our raw materials, and it proved to be up to the task.
That's awesome. Yeah, especially in kind of the area that you work in, Mark, in biopharma. I can expect that you would see such huge successes with the 1064 over a 785 instrument. So I do want to kind of shift then, because, given that the instrument is really created to earn its keep, if you will, in this application of raw material identification. A lot of folks have realized that, but outside of RMID, there are a lot of different applications as well, and our panel has used it for that. Mike, if you wouldn't mind, I'd like to actually pose this question to our audience as well.
Sure, of course. Let's get this question out there. As you can see, outside of RMID, where does handheld Raman have the most real-world pharma utility? And your choices are product development, intermediate formulation checks, brand protection, anti-counterfeiting, screening of finished products, or other. So everyone, please make your selections, and we'll give everyone a few moments. And what we can do is we will gather all of our results.
I am very curious about this one.
And we will push them out. Everyone take a look. So
yeah, I can't vote, so I don't have a voting button.
What would you choose, Joe? What would be your selection?
So definitely, intermediate formulation checks. I have used the unit for clinical complaint samples, and I do know Pfizer uses Raman for anti-counterfeiting.
Oh, yeah.
And we also use it for screening of drug products.
So I should have F, all of the above.
Mm-hmm.
Pretty much.
That's cool. That's really cool. Well, I mean, if you wouldn't mind sharing the results, Michael.
Well, we've got intermediate formulation checks were 25%, brand protection, anti-counterfeiting were 50%, and screening of finished products was also 25%. Do you see the results on your screen?
I think they can. Okay.
Yeah.
And it's interesting to kind of hear that feedback, because really that's how Raman made the leap over from the safety and security realm into pharmaceutical, was kind of making the leap across the brand protection or anti-counterfeit application. So that's very curious. So I will say to the panel then, Joe, I turn it right back to you, my friend. You have definitely put Progeny to work for some really non-standard applications. Would you mind perhaps sharing a little bit about the very unique work that Pfizer does? You're one of the very many large pharma companies that uses relationships with smaller and mid-sized companies to kind of achieve your business goals. Would you tell us a little bit more about kind of how you put Progeny to work in that whole relationship?
Sure. So part of our process is, after clinical drug product supplies are tested, they go someplace to be packaged into the clinical packaging that people are going to see in the clinics, the blisters, bottles, however. And that takes place in most cases at a contract manufacturing organization. It takes time because they have to do the packaging, and then they have to send us representative samples for verification, active versus placebo, and the like. So we've been working with certain partners at the CMOs to perform the Raman testing at the site so that the supplies don't have to come back to Groton or, Sandwich, England, in the case of the UK and the EU. But we can still review the results, their methods that we've developed on the products, and we've transferred them to the CMOs. But it allows us to pick up weeks of time, which is important because when they map out a product all the way from API synthesis to drug product manufacture to signing people up for the clinical trials, everything is on a Gantt chart, and the further along you get, the more delays there can be, but that dose date of that patient rarely changes. So you have to look for all the places you can pick up time. And there are some good products that are friendly, Raman friendly, I call them.
Raman friendly. That's a term that you used before, in talking about this. So that's in a range of API loading, right?
Right. A certain API loading that we've found, usually in the 5 to 20% range, if it's a solid dosage form, that with the 1064 is Raman friendly. We've tried it with the 785 nanometer Ramans, and because of the fluorescence from the excipients and everything, we just can't make any headway with that. It has to be super loaded up with API for us to see peaks above that ski slope of fluorescence.
That's the challenge.
It's not a magic wand. Topicals, super low API loaded drug products or IV solutions. It's not that great, but we've played around with the SIRS technology, trying to push into that area too.
Cool. Very cool. So here is, just a quick diagram kind of how Pfizer works with some of their CMOs on the post-pack ID challenge. So Joe, can you see the progression of this so far?
Yep.
Okay. So would you walk the group for this? Because I learned so much from this when you showed this to me, if you wouldn't mind. Yeah,
no problem. So, the way we look at setting up a program, if it's going to be viable or not with a CMO, is we look at what's on the schedule that we have on the schedule. Are there any of the products on their schedule that are going to be large packaging jobs across the year? If they're one-offs, that may not be so great, but if we're doing 20, 30 of them, and they're good Raman candidates, then we'll definitely zero in on trying to develop a Raman ID method for that product. And
it starts with a business conversation, right? It starts with a business challenge.
Right. We have a flow chart kind of, and it's like, is it worth the effort?
Yep.
It's a good candidate. There's a lot of them, and we can really pick up some time on these. So we do method development in-house, and then we qualify the site. We look at their scans and go through that with them. And if that all looks good, we issue a transfer memo. We update our test method, and then the CMO performs the ID testing. It's either the active or the placebos as part of the packaging process job for us. And then they include the Raman spectra along with the appearance of the dosage form. And that goes into the batch record for review by their QA, and it also gets reviewed by our QA. But we've taken our lab out of that mix, and we can prioritize on samples that do require HPLC or UV or something like that.
That's cool.
And it
And
frees
that shift of personnel that I kind of noted earlier that.
Right. And since we do shift around and, same person that's doing post-pack ID may be doing an API release testing, it gets them into maybe some more value-added testing.
That's cool. And just, again, that communication tool, the way you've also kind of talked to me about the way you've used Progeny and its camera and things to capture, bits of information that are very hard to translate back and forth. And I think that's just really awesome. Keeping with this idea, thanks, Joe, for sharing that again. It's awesome. But keeping with this idea of, making the most of Progeny as it's a handheld spectrometer, right? It's not just a pass-fail tool. Michelle, I know you guys do some amazing things. You've already said you basically do everything with it. But I would love for you to share a little bit more about the work that you do as well, if you don't mind, and specifically about stellates, because it's such a cool technology that you make at Lindra.
Yeah, thanks, Joe. So at Lindra, we are working on developing ultra-long-acting oral dosage forms. So this is kind of a description of the dosage form that we manufacture at Lindra. So it's a pill, and it basically disintegrates and deploys in the stomach. And in the stomach, we have controlled drug dissolution through our stellate, and that's the name that we've given our unfolded drug product. As you can see there, looking like a star. And the stellate remains in the stomach for about a week. And we can tailor the length of time that it's in the stomach. And then basically we have disintegrating matrices that allow the arms to fall off, and that will allow the drug, the stellate, to pass. So when we're using the Progeny, there are many ways and areas that we can use it for, but here's a description of kind of why we would find it helpful. So in our stellate design, we can have multiple drugs. We can have inactive or active arms. And the Progeny is used to both identify the inactive arms, but it's also used to identify the drugs within the active arms. And we've been able to use it to tell mixtures and distinguish mixtures from single drug arms versus a drug A and B mixed together. The other places that we use it are on the full drug product on the coated capsule, so we can scan and identify the components of the coating. And basically, we've integrated it across all of our manufacturing areas. Another nice thing that the Progeny does is it monitors polymorph form. So part of our manufacturing process is to extrude the drug with a polymer. And one thing that we like to monitor is there any changes in the crystal form of the drug after the manufacturing process? And that, because Raman is so sensitive to crystal structure, and the Progeny can be used in that way to kind of monitor if our drug is stable in our dosage form.
That's cool, because literally the extrusion process might change the form, right?
Exactly, yeah.
That's so cool. That's really cool. That's so interesting. Just your entire kind of product and what you do is really interesting. But that polymorph application, when you shared that with me, I was so excited to hear about it. So thank you very much, Michelle, for sharing that too.
Yeah, no problem. It's very cool.
So I want to go back to our audience then and ask kind of another question of you in kind of the idea that we have this panel here. How can they help, right? Let's shift a little bit. Michael, if you wouldn't mind asking the next poll question, which kind of changes focus a little bit to challenges.
Absolutely, definitely. So our next polling question is, what has historically been the most difficult aspect of purchasing or implementing a new analysis tool in our industry? after, of course, the cost. So your selections are writing standard operating protocols, training new revolving team member, change, control, documentation submission, IT, data management issues, or just getting started. So everyone, please make your selections, make your choices. We'll give everyone a few moments and to decide what they want to choose. And we will tabulate those responses right now. And I'm going to show everyone what the results are. So, wow, I hope everyone can see this. Just getting started was the majority, 80%, and training new team members came in as the second most. So I'll hand it back to our panel and Jill to talk about this.
Just getting started. That kind of makes me laugh because I always say it comes down to getting started and just getting a plan going. And I really try to do my very best in helping our customers get over that first big hurdle and get the ball rolling. But I will say then, to our panel, guys, you did it. You put a Progeny into place at your site. What was your first big challenge? Given that you've implemented other tools, what was the first big challenge that you had when you were trying to get Progeny working at your sites? Joe?
So I didn't get the chance to vote, but for me is beyond a doubt IT and data management issues. Getting systems to dovetail with our own data management systems and data archival systems, and again, I'm a broken record, electronic data integrity. We love new technology. It's great. We'd love to try it out. But where we've hit the biggest hurdles is getting that software with the new technology to mesh with our SOPs and what we have to show to demonstrate that data is secure.
Yeah, it is definitely a maze of hurdles. I have not felt your full pain, but just a little bit has been painful enough, my friend. Michelle, how about you? What would you say was a big challenge maybe that you faced?
So for us, I think implementing it has been pretty easy, but really the challenge that we've faced has been determining what is the Progeny most useful for and what are the limitations on the technology. And for example, we receive a lot of polymers that we use in our process, and telling the difference between things like end groups or molecular weight is not something that you can do on the Progeny or with any Raman. And that's kind of just something that we've learned as we've gone on. Where is this, technology applicable versus not?
Yeah, that's it's a good point, and I wish you could. Those long chains are very challenging, but that's scoping it out and realizing where can we have success. That's something I always try to help our customers kind of scope out the problem, focus where you can have the most impact. Mark, you shared with me when we were talking about this prior, you shared with me a really funny early challenge. Would you mind sharing that with our audience?
Well, are you talking about the laser safety or?
No, I was actually when you mentioned to me how everybody wanted to write it in for every method protocol.
Yes. Again, I came at this with very little analytical background, and I found the instrument really easy to use, and gave good quality data right away. I think almost, but the problem with my group, I think, is everybody in the group immediately became convinced that a Raman ID could be done on anything almost instantaneously. So they could come into my office at 3 o'clock in the afternoon and say, oh, we have this new raw material we want to use. Can you do the Raman ID test on it so we can, release it to use in the clinical trial manufacturing area tomorrow?
You had to reel that in a little bit there, Mark, didn't you?
You almost can do it. If you have a substance that that has a good Raman spectra, which is quite a lot of things that I work with do, and if you have a reference material, then you almost can do it that fast. But not everything has a good Raman spectra, and I at least have to order a reference material sometimes and wait for it to come in.
Got to get that traceability in line, right? That's very interesting, Mark. Thank you.
And there's doing the paperwork takes a lot longer than actually doing the test.
Absolutely. So then going back to that last, I think we said getting started, and then I think Mike did the second answer was about change control, right? So that brings me kind of the next point that I wanted to bring up with our panel here, and perhaps you can shed a little bit of light on it, because I do believe it's an area that causes a lot of folks some concern, if you will. But what can you share about your process, maybe Michelle or Joe, Mark, in terms of your approval to get Progeny in place? I know you're pretty early in it again, Michelle, but what have you done?
So, we are early, so we're not in an approval place, but we are kind of, breaching that, being in phase 1-2 trials. Right now, the way that we have been kind of implementing this is building up our library and testing the spectral reproducibility for material ID, and we're just getting started on planning the validation of our methods. So it's, we're lucky because we're early in the company life cycle, and we can build these processes with quality assurance as we're doing it. And that's kind of a nice thing.
It is.
And makes it a little bit easier.
Yeah, it's very interesting to watch you guys build it from the ground up that way. But then, Joe, you're on the opposite end of it. You're in the behemoth with many different ways of doing many things correctly. Kind of how have you approached the process of putting, Raman and validating and that kind of thing?
Yeah, so our SOPs are pretty specific. We group our instruments into different buckets, so typically we would ask the vendor for an IQ, OQ, PQ document. Our QA would have to pre-approve that document for execution. At which point we'd bring the vendor in, they'd perform it. It would get QA signed off again. At which point we start to look at how's the data going to interface with our systems for backup, for cloud storage, all those type of things. And so we might have to write some supplemental test scripts in addition to the vendor's IQ, OQ, PQ, which again gets approved by QA for execution. And then we execute those scripts, hopefully to root out any bugs in the software or anything like that. That gets approved. And then we go through what's called the data integrity risk assessment process, which is a series of questions that we have to answer. And that, again, looks to root out any other kind of potential blind spots if we get audited or something like that. So we go through that. That gets QA approved again. And then at that point, it gets put into our calibration system and program, and it's officially labeled as a GMP instrument. That's just the qualification, nothing to do about the validation. We have different things we do about that too. But
okay, that's its own mountain of work too, right?
It is, but it's not bad. It's a lot different when you can collect a scan in, probably less than 30 seconds to validate something versus setting up a whole HPLC run or on any other instrument. So the validation that's pretty much the easy part. It's getting it to that GMP sticker on it. Right.
The magic golden star, right?
Right. Right.
I know that sticker. I know that Pfizer label. It's a metal sticker.
It is. Yeah.
So I will turn then to the next part of the presentation here, and it really does. It's a $1 million question. If you could start over, given hindsight is 20/20, what would you do differently? I'd love for you to share it. As crazy as it might be for me to ask, what would you do differently, Joe?
I think I would pay attention to how the software interacts with our internal systems more. I think we stumbled into some things when we were trying to qualify it that we really didn't think about when we first had the instrument in, and we threw all these compounds and stuff at it, and it was just cranking out spectra. It looked great. We didn't think down the road how is the data going to get off the instrument? How is it going to be backed up, and how did that work with our own internal restrictions that we have on permissions, on folders, on security settings, on the PCs, right? I mean,
yep.
We have a ton of them, right? I'm doing this on my personal PC because my Pfizer one, it's got too many. Yeah. So, but that being said, it was kind of a perfect storm. When we got the Progeny in, it was just when the whole data integrity microscope, when auditors were really starting to look at data integrity. So, 5 years earlier, it would have been a lot quicker. But because of the added emphasis on data integrity and what auditors are looking at, we really had to make sure everything was bulletproof.
That makes sense.
And that's FDA, that's MHRA, all of them.
Yeah, no choice, right? So that kind of it does also make me think about where teams generally get stuck. Michelle, would you share anything too about kind of hindsight is 20/20? Does anything stick out at you as something you would do drastically differently so far?
To be honest, Jill, I don't have anything that I would do very differently, just because we are still kind of building our systems. And I think if anything, it would be kind of echoing what Joe said, and like understanding how the software and transfer and copying the data. It's understanding that and how because we're building our quality systems right now, and it's really kind of an unknown area for us. So kind of understanding where to store the data when you download it and that sort of thing is more what we're looking at.
Well, maybe Joe and Michelle, you can share some advice directly with each other, certainly. But that is definitely the case where, we see teams certainly get stuck in kind of the same areas over and over. So scoping out that problem and making sure that you have the appropriate samples, those are always two of the main steps that I kind of encourage our customers to address for themselves, because proper planning always prevents poor performance, the 5 Ps. I would say, Joe, you also said that in some conversation that making sure that different groups within your organization had an idea of how to use the tool is something that you've gone to great extent to make sure that is in place. And then Mark, I know you're on the line. You talked also about making sure that your laser safety group was involved early on in the process too. That would be something that you'd give some advice around, right?
Yeah, the regulatory environment for lasers in Massachusetts, I guess, changed from when we first bought the instrument and started using it. And then, about after using it for two or 3 years, the safety group here came to me and said, we have to upgrade we have to talk to you about some of the safety issues involved in using the laser. And even I'm getting a new lab built, and right now the safety people are holding up the finishing off the new lab because they're not satisfied with the laser safety stuff involved with it, having locks on the doors from the outside, and
it's always important to make sure that we're involving all those team members, right? Because everyone's got some different perspective of how to approach things, and
we didn't budget anything for laser safety, so we had to go back and ask many people for some more money.
So I will then we're getting close to our time for today, folks. I wanted to kind of finish up by asking our panel here to be progeny gurus, if you will, for the quality professional looking to bring a handheld Raman into their organization. What's the 10-second, 20-second, 30-second advice that you'd give them, guys?
Well, I'll go first. So ease of use, user-friendly, intuitive, and again, a really robust software as far as how the data is handled.
So you say that you're referring to Progeny, right? Or those are goals?
Any system that comes in, but yeah, Progeny in particular.
Michelle, would you kind of give out any advice that we haven't covered yet here today?
Yeah, I think understanding, that you can really have modular control over things like understanding that you can really work on the collection parameters that, on the progeny itself. So the power and the exposure time and, understanding your sample needs.
That's interesting. So playing with the actual scanning parameters that's cool. Yeah, I appreciate that too, given I have experience with different handhelds, and given what Progeny can do, I really appreciate that about it too. And then Mark, I want to end with you. Do you have any final advice that you might want to share about Progeny or about our team here at Rigaku before we close out?
Well, I was really surprised at that poll question that you asked, and a lot of people said the hardest thing was getting started. I mean, I found it very easy to start with and to use and everything, and I am talking about the Progeny, and
that's cool.
I would just encourage people to try it. If their experience is anything like mine, I'm sure they're going to be very happy with the results they're going to get, and for without taking up a lot of time either. And actually, I've been helped by the people at Rigaku quite a bit. So, if they had any problems, I would encourage them to contact you guys, because you've been very helpful to me. So
I'm glad. Thank you, Mark. I'm glad. We like to be helpful, that's for sure. And we have very little time left here today, but I definitely want to make sure that we're addressing any questions that did come up. Certainly, if you did ask us any questions, we will make sure to follow up with you. But I want to say thank you so much, so very much, Joe, Michelle, and Mark. I can't see you, Mark, but I know you're there, and I'm so grateful for you joining us today. This has been a really fun and interesting conversation for me. Certainly, thank you to APR and to our attendees as well. It's been such a pleasure. I'll turn it over to Michael Auerbach from APR for moderating our Q&A if we have some time.
Yeah. Thanks, Joe. I think we have, well, first of all, I'd like to thank all of our panelists for their information and their sharing their experience with us. And if anyone has any questions for anyone, to please type them in right now. We do have a question here I'd like to present, so let's do that. Our question is, what do you recommend as the preferred method of annual recertification?
Okay.
So the calibration, you mean? Annual calibration? Is
I
that?
believe, yeah, recertification of the instrument.
Yeah, we do that at Pfizer. We do that in-house. It's if we're talking about the annual calibration, we calibrate it on an annual basis. It's very easy. It takes, I don't know, maybe a couple minutes, but we do that in-house, and so that's it's pretty much a non-issue.
We use our remote option, right? So to answer the question as completely as possible, Michael, we do offer 3 options for that for our customers. Joe and Pfizer use our remote annual recertification option. So they get a document and they perform the procedure, and then we take a look at those results and issue a certificate. But we can also do that in-house in our facility or at-house at your facility as well, depending on what your company prefers. So,
okay, all right. I think that's all the questions we have right now. So I'd like to thank everyone, all of our participants, for sharing their knowledge with us, and also offer a special thank you to Rigaku for sponsoring today's event. I'd like to mention to all of our attendees to keep a lookout for an email containing a link to view this webinar on demand that you can review the webinar and also share it with your colleagues. So again, thanks everyone for attending, and please enjoy the rest of your day. Thank you.
thank you so much.
All right.
Have a great day, guys.
Bye-bye.
Okay, thank you. Bye now.
All right.
Thank you.